Hello, my name is Rosalind Sadleir, and in this presentation I will describe some findings from our recent MRM paper, which is titled Functional Magnetic Resonance Electrical Impedance Tomography Sensitivity Analysis Using an Active Bidomain Finite Element Model of Neural Tissue. This work was carried out by our team in the School of Biological and Health Systems Engineering at Arizona State University and describes modeling work intended to establish the feasibility of the fMREIT technique to directly identify active neural tissue. There are a number of methods that can be used to non-invasively image neural activity. These include blood oxygenation level dependent or BOLD fMRI, inverse electroencephalography EEG, and magnetoencephalography or MEG. BOLD fMRI is widely used to image activity with higher spatial resolution than inverse EEG or MEG. However, it is less temporally direct, and timing information must be deconvolved from signals using the hemodynamic response function. Direct imaging of neural currents using MRI or NC-MRI may be difficult because of the small signal sizes, as well as signal cancellation that may be caused by multiple overlapping and differently oriented sources. During neural activity, membrane conductance changes, and the apparent conductivity of active cell regions should also change, as shown here on the right. The white lines here show current streamlines passing around cells with lower conductance in active membranes. Because the low-frequency current can't penetrate the cells easily, the entire cell looks like it has a low conductivity. If ion channels open and an action potential occurs, the membrane's conductance increases and current can flow inside the cell. The current pathways are affected as a result. MR phase images that are recorded during current flow encode the Z component of the magnetic flux density caused by the external current, and also register these shifts in the current flow that may be caused by activity. The amount of phase change measured should depend on how much external current is administered and also on internal source strengths, sizes, and locations. This technique may be useful to enable direct imaging of activity. Because the imaging currents may also affect activity levels, the contrast might be usefully exploited to image activity caused by neuromodulation techniques such as deep brain stimulation or transcranial DC or AC stimulation. We recently demonstrated an activity level dependent contrast in fMREIT using in vitro tissue tests. This work was reported in the journal Neuroimage. We used a finite element model to explore the feasibility of the fMREIT technique and determine typical signal sizes. The model was created using COMSOL multiphysics. A 3D sample chamber is shown here in cross-section and from an oblique view. The octagonal sample chamber had 8 recessed electrode ports, and the active tissue was surrounded by a bath of saline solution. Because this model was based on our in vitro experiments using the abdominal ganglion of the aplysia, we used conductivity properties and dimensions typical of these experiments. The active tissue of the abdominal ganglion is the cylinder you see in the center of the sample chamber. The active tissue was modeled using a bidomain representation, comprising averaged intracellular and extracellular regions connected by an active membrane model that was based on the Hodgkin-Huxley equations. More information on the model specifications can be found in the supporting material for this paper. We located an isotropic source of activity in the center of this active tissue. The amount of current-related phase signal encoded in an image depends on how much current flows during the sequence echo time. In the spin echo MREIT sequence shown here, we inject a positive current after the 90-degree RF pulse and the opposite current after refocusing in order to preserve the signal. In many cases, we repeat an image using the same sequence but with a negative going current pulse just after the 90-degree. These two images may then be subtracted, which produces a composite image with twice the amplitude and with any systematic background phase. These plots show MREIT imaging currents after the 90 and 180-degree pulses. These are noted with A and B, and they're both shown here as positive, and they're placed against a background of pulses in the internal source current. The effects on the phase images of the conductivity-dependent contrast will be greatest if any activity is happening as the imaging currents are applied. In the panel below, you can see how the active bidomain model responds to the activity in the internal source and show. These images show initial results from the model. At the top, we have an image of the Z component of the magnetic flux density caused by a 1 milliamp current flow from the top right electrode to the bottom left electrode. The active tissue border is shown with the dotted line. Because we are also interested in the effect of the imaging current on activity inside the tissue model, we also plotted the current density distribution caused at the peak of the imaging currents. This measure can broadly be used to predict how likely it is that activity will be caused. These images show simulated MREIT phase images that have been rescaled to BZ and integrated over the echo time of the test spin echo sequence. The leftmost images show the effect on phase of the internal source activity alone and are equivalent to NC-MRI images. We used two isotropic source strengths, 500 picoamp and 2.5 nanoamp, respectively. The second and third columns show how the effect of changes inside the active tissue. This slide shows another view of the data in the images from the previous slide. To be able to see the effects of the activity-related phase changes must be above the noise floor. In the ultra-high field imaging sequences we use at present, the noise floor is around 0.1 nanotesla. Here we are showing histograms of the phase of differential BZ data relative to the noise floor. The numbers on top of the noise floor measures show the number of averages that would be needed to achieve this noise floor at resolutions. Activity locations can be better isolated using the Laplacian of these delta BZ images, which is what is shown in this image. Because the simulated data doesn't include any noise, the images look similar in each case and the imaging current effects are disappearing. The central area is larger at the larger internal source strength. Here is a close-up of the same image. You can see both the source and the edge of the active tissue here. The edge of the tissue is visible because of counterflows caused by the activity in the center. The Laplacians of the delta BZ data can be used to reconstruct the Laplacians of conductivity distributions using an MREIT reconstruction method, which is called the Harmonic BZ method. This method requires knowledge of the voltage gradients formed by the imaging currents. We cannot perform a harmonic BZ reconstruction for the no current data on the left, but we can show reconstructions of the Laplacians of the conductivity changes for the two MREIT cases. Noise was added, and here is a close-up of the reconstructed conductivity change Laplacians again, showing the effect of that added noise. Because computing Laplacians intrinsically adds noise to the data, it is more likely that analysis of phase images will be done comparing one task with another or a task with rest using similar analysis methods as are used at present in bold fMRI analysis. This final slide gives an idea of the interplay of the external MREIT imaging and the internal intrinsic current sources and demonstrates how we see larger signal changes with larger external imaging currents. Also, that there are saturation effects with larger internal source strengths. Finally, we also see an indication that in one case with 1 milliamp external imaging current, there was a decrease in the median change in BZ data because the imaging current depressed activity in the active tissue part of the model. Our model results indicate that it should be possible to use fMREIT and moderate imaging currents to image neural sources as long as noise levels can be made low enough. Because the size of signals depends on imaging current amplitude, we hope this method has promise as a way of imaging neural activity caused by neuromodulation currents using these neuromodulation currents as MREIT sources. This work was performed in the Neuroelectricity Laboratory at the School of Biological and Health Systems Engineering at Arizona State University. We thank all our lab colleagues. This work was made possible with the assistance of funding from the National Institute of Neurological Disorders and Stroke.